Hello children, I'm Ravi Chandra from Sri Chaitanya. Hope you're doing great. Today we'll be discussing a chapter called Plant Growth and Development. So it is nothing but, in short, the success story of a plant from a very tiny seed to a huge giant plant. How it grows and how it develops. We'll explore the entire story without wasting any time. Right. Now, the first important is what is the meaning of development? All the precise and orderly succession, both promotory and inhibitory. Now the important point here is development not only include promotory but also the inhibitory changes taking place in a plant is said to be development. So shall I say that? Does it include growth? Yes. Does it include differentiation? Yes. So simple way of saying development is it includes both growth and the differentiation process. Now development include, question is, I said it includes both promotory and inhibitory. What? From seed germination to senescence. Simple way to remember, S to S. Seed germination to senescence is said to be development. What are the changes? First, does it include seed dormancy? Yes. Does it include germination of seed, which is the first step? Yes. Does it include seedling formation? Have you ever seen the sprouts of bean, children? You see a small white stalk developing out like this. Have you ever seen this? Yes. This is said to be a seedling. Seedling. Anything which is yet to become green and a tiny development from seed after its germination is said to be a seedling. So seedling production is also development. Plantlet, if it is green, you call them as plantlet. It is also development. Young plant producing roots and stems and leaves, development. If it becomes mature, producing reproductive organs, it is development. If it is showing degrowth, where catabolic rate is more when compared to anabolic rate, senescent, it is also development. Question is, does death a development? No. From seed germination to senescence, only these are called development. Next, plant part shows both indeterminate and determinate growth, children. Root, stem and leaf are vegetative organs. Flowers, fruits and seeds are reproductive organs. And all of you know that root and stem shows indeterminate growth because of apical meristems. Then leaves, flowers and fruits are going to show determinate growth because there doesn't have any apical meristems at its tip. Why vegetative phase should be prerequisite to flowering? Very simple, for two reasons. One, the plant requires some time to gain maturity. Two, the food is produced during vegetative phase and these food liberate energy and that is very much needed for flower, fruit and seed. And the transition from vegetative to flowering place is under the influence of light, temperature and hormones. This is not there. The effect of temperature on flowering is vernalization. The effect of light on flowering is photoperiodism. In living world, seasonal breeders require a certain photoperiod for reproduction. That word is given there. There is a direct link between the structure of cell tissue and the functions they perform, correct? And if root hairs, they help in absorption. Parenchyma, this tissue, help in storage. Collenchyma, flexibility. Sclerenchyma, mechanical strength. So there is a link between the structure of cell, lignified walls, and the function. Only when they have lignified walls they can provide mechanical support. Then this diagram is important, children. Apigeal germination. What is the meaning of apigeal germination? Look here, this is the most important part. This is the radical and this is called hypocotyle. You know what the meaning of hypo means? Below. It is present below the two cotyledons. You could see the two cotyledons. This axis is present below the cotyledons. Therefore, it is rightly called hypocotyle. See what is happening, children. This hypocotyle is elongating. It's further elongating. And when this hypocotyle elongate, The cotyledon, look here, the cotyledon is brought above the surface of the soil. The only doubt you have is why the seedling is curved. Because if it is straight, then it has to encounter what is called the resistance from it. So it wants to protect its tip. So therefore it's going to show a small curve. Then it is going to go up, and the cotyledons are brought above the surface of the soil. This is called epigeal germination. So it's very clear. It is the hypocotyle which is elongating and bringing the cotyledons above the surface of the soil. Then what is epicotyle? This is epicotyle. All right. Your textbook called this as epicotyle hook. This part is epicotyle hook. Strictly speaking, it is actually a hypocotyle hook. Let us pray that this question doesn't come. All right. The textbook says epicotyle hook. Go according to epicotyle hook only. But actually it is the hypocotyle which is forming a hook-like structure. Could you tell me what is the hormone responsible for this? Ethylene. Ethylene is responsible for apical hook formation. We discussed about that. So this is very simple, children. So these are the two cotyledons. If I just explain you. All right. So this is the radical. This is called hypocotyle. This is cotyledonary node. This is epicotyle, and this is the plumule. So listen, when the hypocotyle elongate, the cotyledons are brought above the surface of the soil. So what you see in bean is the elongation of this. So then we talk about some important and simple points about growth, children. These we already discussed already. Growth is an irreversible. This is important. Why sponge is not a growth? When you put it in water, it is going to swell. Why you're not calling growth? Because again, it loses water. Growth means an irreversible process. It is a permanent increase in size of an organ. Growth occurs when anabolism is more when compared to catabolism, naturally. When the leaf is going to become old, catabolism will be more when compared to anabolism. That results in degrowth or senescence. Remember that. Non-living things, can they grow? Yes. They increase in size due to the accumulation of materials on it, extrinsic. Does sand dune grow? The answer is yes. Does sponge and wood grow? The answer is no. Because in sponge and wood, it's an reversible phenomena. In sand dune, it's an irreversible phenomena. Therefore, sand dune shows extrinsic growth. Wood and sponge doesn't show any growth. Then we talk about primary and secondary growth, children. For that, I want to show you a simple diagram here. These are apical meristems. These are intercalary meristems. All of you know that. Apical meristems and intercalary meristems are primary meristems. They divide and redivide and help in the increase in the length of the plant organ. You know pretty well, after some time, these cells lose the power of division and undergo differentiation and give rise to primary permanent tissues like parenchyma, collenchyma, sclerenchyma, primary xylem, and primary phloem. What is this process called? Differentiation. Simple way, I'll tell you. Different types of permanent tissues are formed by differentiation. Different types of permanent tissues are products of differentiation. Different differentiation. So easy way to remember. This type of growth, where the plant is increasing in its height, is said to be primary growth. Apical and intercalary meristems play a role. And you know these are vascular cambium and cork cambium. A circle, if you take a longitudinal section, surely it will be in the form of two lines. Isn't it, children? Vascular cambium and cork cambium. In transverse section, they are circle. In longitudinal section, they'll exist as two separate lines laterally. That is the reason why it's called lateral meristem. You know that vascular cambium produce xylem and phloem. Cork cambium produce cork and secondary cortex. It means that there is an increase in diameter because of lateral meristems. And this is said to be secondary growth. So initial growth is primary. By the activity of cambium, increasing in circumference is said to be secondary. Well, you know that, increasing girth. So what are the points I've written here? Here I have written some important points, children. Plant shows unlimited growth due to the presence of meristems. Where they are present? They are present at root apex, shoot apex, at the nodal regions of grasses, and also in the lateral regions. These are called lateral meristems. These are called apical meristems. This is intercalary meristem. Products of meristems lose the ability and become permanent. This process is called differentiation. Such differentiated cells make up the primary plant body, which include all these tissues. This is simple to remember. Then what is the difference between primary growth and secondary growth? Primary growth is seen in all plants. Root tip and stem tip is the location of those meristems. Type of growth is increase in length. Meristems involved in primary growth is apical and intercalary. It occurs in the early stages of plant, helps the plant to grow tall. Then next one, what is secondary growth? Seen in dicots and gymnosperms. It is by the activity of vascular cambium and cork cambium. It is increase in the girth or circumference of the plant organ. All right, and it occurs in the later stages, not in the early stage, later stage. It helps in increase in the girth, mostly in root and stems of dicots and gymnosperms. The next topic is measurement of growth at cellular level. The correct definition of growth at cellular level, children, is increase in protoplasm. But can we measure the increase in protoplasm? It is very difficult to measure. The reason is very simple. Cell itself is small, and the protoplasm is a part of such a small cell. Understanding its enhancement is a very difficult job. As a result, not only that, the protoplasm is very dynamic. It contains plenty of water. It keeps on changing. So therefore, increase in protoplasm is very difficult to measure. Therefore, we found out some other parameters which are equivalent to increase in protoplasm, which can be easily observable. So let us see what are those characters which you can say. Indirect parameters could be fresh weight, could be dry weight, could be length, could be volume, or could be cell number. Increase in cell number, maize root tip, all right, 17,500 cells. Increase in cell volume, watermelon fruit, almost 350,000 cells, 350,000 times. Increase in length, pollen tube. Increase in surface area is leaf. So by looking at increase in surface area, increase in length, increase in cell volume, you can easily what is called summarize that there is an increase in protoplasm. That is very easy one. Next, we talk about phases of growth. Children, this we already discussed in morphology chapter. At the root tip, you see meristematic phase, and you know the characters of meristems. Cell wall is thin, plasmodesmata are abundant, protoplasm is dense, nucleus is large, vacuoles are small, they undergo active cell division. That is the extreme tip. This is the what is called the meristematic zone, all right, or the initial phase. Then far away from root stem junction is said to be distal. This is far away from root stem junction, therefore distal. This is nearer to root stem junction, therefore it is proximal. Then we talk about the elongation phase, this region. All right, here the cells are going to take water. They become turgid, all right, and they elongate. So the cell wall is relatively thicker. Plasmodesmata are present but less. Protoplasm is peripheral because of large central vacuole. This is important. Why large vacuole? Because more water enters into the cell and nucleus is small. Then the next phase is maturation, this part, from where you see the lateral roots develop, all right, and even root hairs develop. That was the question which was asked in the NEET exam. All right, maturation. Plasmodesmata are less, but protoplasm undergo modifications. All right, vacuole is large and single, and the cells are differentiated and then they become permanent, performing a specialized function. Maximum protoplasmic modifications, maturation. Maximum vacuole size, elongation. Maximum plasmodesmata, dense cytoplasm, large nucleus, meristematic phase. So these points are important. Parallel line diagram. Look at this, children. This is the region of elongation. Look at the distance between these lines. It is more, amply signifying that there is more elongation or more growth here. The parallel line diagram clearly suggests, all right, it shows maximum increase in space in between parallel lines in elongation zone, proving that this is the region where maximum growth occurs due to turgidity. Next, we talk about the next important unit, children, that is said to be plant growth rate. What is the meaning of growth rate? Listen, the meaning of growth rate is when you talk about growth in terms of time, then you call it as growth rate. So you're not only talking about growth, but you're also bringing time as a criteria. So both for arithmetic and geometric, if you observe the graph which is given in your textbook, the x-axis is always time. So when you measure growth of a plant organ or a plant with respect to time, then you call it as rate. So there are two types of growth rates, children. One is arithmetic growth rate and geometric growth rate. What is arithmetic? The name gives. It is, first of all, let me show you what is arithmetic. Then you'll understand it properly. All right, look here. I don't have time to draw it, okay? Please concentrate what I've written here. This is the meristematic cell. It divides and produces two daughter cells. One daughter cell is going to remain meristematic, the other become permanent. Means after one division, the number of permanent cell is 1. This is the one permanent cell after one division. Then this cell again going to divide. When this cell again going to divide into 2, then this cell have become permanent. So after 2 divisions, the number of permanent cells are 2. Again this cell divide into 2. This cell have become permanent. After 3 divisions, the number of permanent cells have become 3. And after 4 divisions, the number of permanent cells have become 4. So it's very clear by looking at this diagram that the increase is more or less uniform. Look here, the increase, with increase in time, the increase is more or less uniform. This type of growth gives a linear curve, a linear line, and this is said to be arithmetic. You heard about in mathematics 1, 2, 3, 4, this is arithmetic proportion. This type of growth is seen. Now that is arithmetic. Let me explain you what is written here. Arithmetic growth is a linear growth. All right, where the daughter cells continue to divide, one daughter cell continue to divide while the other cell become permanent. That is the one what you see. One daughter cell continue to divide, other become permanent. Growth is constant and linear. Straight line on plotting, increase the length. Look here on x-axis time, height of the plant. Examples for arithmetic growth is root elongation and the stem elongation. All right, it is more or less uniform. It doesn't occur abruptly, isn't it? You have observed. So that is what is called arithmetic growth because few cells are becoming permanent. Permanent cells added up, all right, in a systematic way. Then what is the examples? Root elongation, stem elongation, and lateral stages of embryogenesis are examples for that. You could see that the cells are dividing like this, we have talked about. Now, what did I say? Root and stem exhibit geometric growth at cellular level. Some of you got a doubt. At root tip, you said meristems are present. Meristems, every cell of meristem will divide. Then it should be geometric. Of course it is. But if you talk about the entire organ as a criteria, then it should be arithmetic. So if they ask you, root apical meristems at cellular level shows geometric, because just like embryo, every cell will divide. But after some time, one cell become permanent, undergo differentiation, means it adds cells, permanent cells in a uniform manner. That type of growth is said to be arithmetic. So if they ask you root and stem elongation, your answer should be arithmetic, children, even though the root and shoot apical meristems at cellular level shows geometric growth, but elongation of root and stem is more or less uniform. It exemplifies arithmetic growth is something which you have to remember. All right, the equation for arithmetic growth is LT, means after time t, L0 initial length plus RT. This is the rate per unit time. So I gave an example here, children. Suppose a root is 5 centimeters. All right, so LT is equal to, LT you have to find out, sorry, L0 is equal to 5. Then grows at a constant rate of 2 centimeters per day. 2 centimeters per day. Here I have written already. All right, so 2 centimeters per day. time is 4, R is equal to 2. So you know that LT is equal to L0 plus RT. So L0 is 5 plus rate is 2 centimeters for how many days? 4. So 4 to the 8, 8 plus 5, 13 centimeters. So after 4 days, the root length will be 13. Initially it is 5, later it is 1. So if I just draw a graph, it will be like this. All right, so this is the proportion. So the number of days, this is the number of days, and this is the increase in length. So you could clearly see after 1 day 7, after 2 days 9, after 3 days 11, and after 4 days 13. So 13. So therefore you could easily tell the number of days here and the length here. That is said to be a linear curve. That's absolutely simple. Then we talk about geometry growth. Then I told you what is the meaning of geometry growth. Here every cell will divide into 2 children. Again this cell will divide into 2. Again this cell. 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 No question of any permanent cell forming here. So therefore first 1, then 2, then 4, then 8, 16, 32, 64, 128, 256, 512, 1024. So like this, the number of cells keep on increasing because every cell is dividing without becoming permanent. All right. So then the shape of the curve is a sigmoid curve. Initially the number of cells are less. Therefore you see a lag phase. Then suddenly all the cells, because they are dividing, all right, then it is said to be log or exponential phase. But after sufficient number of cells formed, the nutrient deficiency and the conditions are not viable for the cell to grow further, then it reaches a stationary phase. This entire curve is said to be a sigmoid curve. All right, but when we talk about geometric growth, this actually signifies what is called the geometric growth, where every cell is dividing and increasing what is called in its size. But after some time, it is going to become S. Actually, geometric is J shape, but in natural conditions, it doesn't exist. Because of that, it becomes sigmoid curve. So that the living cell under natural conditions is going to show a sigmoid curve. And this is said to be what is called geometric growth proportion. Lag phase is slow. Log phase is geometric growth or exponential phase. Stationary phase, growth slows down further or stops. Example for geometric growth, children: bacteria or the cells in culture medium. Now don't take this because you'll get confused again. All right, leaves, flowers and fruits under natural conditions. Initially the leaf is small. All right, then it is going to increase in its size, surface area. All right, then it is going to show a senescence or degrowth. So it's a wonderful example of what is called a sigmoid curve. Plant and animal growing in natural environment. It is the same, children. Plants initially, all right, shows less growth. Later all the plant organs are going to show more growth, then they become senescent. So again you see a sigmoid curve. Early part of embryogenesis, every cell is dividing. Therefore it's also an example for geometric growth. During embryo development, initial growth is geometric and later the differentiation starts, then it becomes arithmetic. That's what we see. You could see the geometric growth here followed by arithmetic. All right. Most plants show sigmoid growth curve. Means growth is not purely geometric throughout, as conditions may turn limiting. Hence it is S-shaped, not J-shaped. All right, then the equation for that is Wt is equal to W0 into e power RT. All right, so let us give one example here, children, with regard to R. A plant embryo has initial weight of 10 mg and it grows at a relative rate of 0.2. All right, what is the mass after 5 days? So you know that Wt is equal to W0. What is W0? W0 is 10 to e power RT. R is the rate, this is not minus. All right, 0.2 into T. How many days? 5. So 0.2 into 5, it comes to 1. So it means 10 into e power 1. So the e value, all right, it's a mathematical value. It's not a biological value. That is 2.7. So therefore, that means it comes to 27 mg after 5 days. This is a wonderful example of how the embryonic cell is going to grow in a geometric proportion. Then we talk about absolute growth rate and relative growth rate, children. In your textbook, they gave an example. A leaf, 5 have become 10. What is the absolute growth rate? The absolute growth rate means the total amount of growth. That is 5 has become 10. So the absolute growth rate is 5. This is easy to remember. All right, final minus initial. Then there is one more leaf they gave. All right, 50 have become 55. All right, here also the absolute growth rate is also 5. Final minus initial. Then what is the relative growth rate? Relative growth rate means you are taking the growth in comparison to the initial size. 5 have become 10. So it means that it has increased 100%. All right, then 50 have become 55. It has increased only 10%. Understand? Absolute growth rate for both the leaves is same, but the relative growth rate of smaller leaf is more when compared to the relative growth rate of larger leaf here. It is also true that sometimes the absolute growth rate may be different, but the relative growth rate can be same. Absolute growth may be different, but relative growth rate may be same. Imagine a leaf of 5 centimeters become 10. What is the absolute growth rate? All right, and what is the relative growth rate? If you look, 5 have become 10 and 50 have become 100. If you understand, the absolute growth rate is what is called here is different. 5 have become 10. So the absolute growth rate is 5 and the absolute growth rate of this is 50. Absolute growth rate is different, but the relative growth rate in both is 100% and 100%. So it means that the relative growth rate is same, absolute growth rate is different. But in this case, we talked about absolute growth rate is same, the relative growth rate can be different. So that is an important point to remember with regard to absolute growth rate and relative growth rate. Then we talk about the conditions for growth, children. Water is required because it helps in turgidity. Nutrients are required because it builds protoplasm and produce some respiratory substrates as well, which act as source of energy. Oxygen, it helps in releasing energy from respiratory substrate. Temperature has an effect on enzymes. Light has an effect on phototropism and also on flowering. All right, gravity. So the root is going to grow towards the gravity and the stem is going to grow against the gravity. All right, so the gravity also have a role to play with regard to its movement. Then we talked about the concept of differentiation, de-differentiation and redifferentiation. Already I told you, when the primary meristem, children, when the primary meristem is going to give rise to primary permanent tissue, this process is called differentiation. Differentiation. What is the next one? Primary permanent tissue, when it becomes secondary meristem, then this is called de-differentiation. So when the permanent tissue remembers its flashback, long back, I'm a meristem, and if it becomes meristem, that process is said to be de-differentiation. When this secondary meristem again lose the power of division and becomes secondary permanent tissue, then it is called redifferentiation. This is nice, I told you. When the primary meristem give rise to primary permanent tissue, it is differentiation. Primary permanent tissue, when it gains the power of division and become meristem, then it is de-differentiation. When such secondary meristems lose the power of division and become permanent tissue, then that is said to be redifferentiation. All right, then some of the examples we are going to read, children, here. Let me see the same definitions I told you. What are the products of differentiation? Simple. The entire primary plant body is a product of differentiation. So therefore, epidermis, root hairs, trichomes, guard cells, subsidiary cells, bulliform cells, hypodermis, general cortex, endodermis, pericycle, primary xylem, primary phloem, fibers, collenchyma, medulla, primary medullary rays. All of them are seen in the primary plant body and they are permanent cells. Therefore, they are differentiation. Then products of de-differentiation. What is the meaning of de-differentiation? I already told you. When permanent tissues regain the power of division and become meristematic, it is redifferentiation. All of you know, in a cambium, these are the vascular bundles. This is intravascular cambium. Children, some of the cells of medulla, they remember their flashback and become meristematic. Look here, This is said to be what is called the product of dedifferentiation. And some of the cells, look here, some of the cells of cortex undergo dedifferentiation and is going to give rise to cork cambium. So the cork cambium here and interfascicular A and B, these are the two examples of dedifferentiation. All right, interfascicular cambium, cork cambium, tumor formation, tissue culture, they're all the products of dedifferentiation. Then this is then the products of redifferentiation. Already I told you, this vascular cambium, all of you know children, this vascular cambium cuts off new cells internally called secondary xylem, externally called secondary phloem. This cork cambium produce cork externally and secondary cortex internally. Is cork, secondary cortex, secondary xylem and secondary phloem permanent tissues? Yes. Are the secondary permanent tissues? Yes. Therefore, they are the products of redifferentiation. That's what I've written here. Products of redifferentiation. Secondary xylem or wood, secondary phloem, cork, complementary cell, secondary phloem, periderm include all these three. So therefore we say that in periderm, cork and secondary cortex, those are the points which are the products of redifferentiation. Then I asked a nice question. Tracheids and vessels in xylem can be the products of differentiation and redifferentiation. If tracheids and vessels are seen in primary xylem, differentiation. If tracheids and vessels are seen in secondary xylem, that is wood, redifferentiation. That looks very simple. Now I'm pretty sure that you understood all these points. Plant growth is open because it could be determinate or indeterminate. Not only plant growth is open, even differentiation is open. What is the meaning of open differentiation? Cells arising from same meristems can possess different structures at maturity depending on their need. So therefore remember, both growth and differentiation are open is an important point to remember. Then we talk about the final structure of cell after differentiation is determined by the location of cell. You know pretty well about that. Then differentiation results in development of structures that commensurate with the functioning the cell finally perform. Such differentiation is open and the development is also flexible. These are all the points which you know pretty well. Then these are the points we already discussed, children. All the changes, both promotory and inhibitory, from seed germination to senescence is said to be development. Development include both promotory and inhibitory effects. Then we talk about the development in a cell. We'll be discussing about the development occurring in a cell. First, look here. This is a meristematic cell. There are two options for meristematic cell. Either it can show increase in protoplasm and can divide. This is option number one. It keep on dividing and become a meristematic cell. Option number two is this meristem can lose the power of division and undergo cell elongation. This is B, cell elongation. After cell elongation, then it can differentiate, and then in that process it is going to become mature, and then it is going to become senescent. If you observe this graph carefully, children, there are two options for meristematic cells in its development. Either it can divide or it may lose the power of division. When it divides, it is shown like this. When it loses the power of division, the first thing it does is it's going to elongate. Then after that, it is going to start performing a specific functions, undergoing maximum protoplasmic modifications, maturation. All right, this after elongation, the cells are going to differentiate and then mature. So it means that after elongation, the cells get into this maturation phase. This process is said to be differentiation. That is the reason why this line is given on the top. This phase, this transition from elongation to maturation has to pass through differentiation. What I mean to say is that after the cell expands, it will differentiate and start performing a specific functions you call such as a maturation phase, and then it undergoes senescence. This is very easy one. What did I write here? The cell exhibiting plasmatic growth can either divide again or lose the power of division and undergo cell expansion and later undergo differentiation and maturation during development. That looks very simple. Next, the last part in this topic is plasticity. What is the meaning of plasticity? Ability of a plant to change its growth, development or form according to environmental conditions or different phases of growth. That is said to be plasticity. Ability of a plant to change its growth and development process. Wonderful example for that is heterophily. Different shapes, either due to its development or due to environment. Look at this in cotton, larkspur and coriander. In the early stage, the leaves have with less number of lobes. In the adult stage, the leaves have more number of lobes. This is a wonderful example for developmental heterophily because it occurs in its life cycle. Then the next type of heterophily is environmental heterophily. Look at this. This is a leaf which is present aerially in a buttercup. Buttercup is the stem is partly submerged and partly aerial. From aerial part, the leaves are normal, but the submerged leaves, they are dissected such that they can easily float in water and can even absorb water. So therefore, this type of heterophily is because of two different environments. Therefore, in buttercup, that type of heterophily is said to be environmental heterophily. Look here, these are different shapes. The leaves in young plant is different to mature plant in its life cycle. Example, cotton, coriander and larkspur. So the larkspur scientific name is Delphinium, cotton is Gossypium. Submerged leaves are dissected and aerial leaves are normal. All right, they differ in shapes. Again shapes. All right, because of two different environment. The scientific name of buttercup is Ranunculus. That completes, children, the first part of the topic, which is said to be the plant growth and development, which I'm pretty sure that you understood every word which I've discussed here. Then we talk about plant growth regulators. We get into this part. This part is more of remembering. You have to find some ways to remember. I'll help you out in some aspects how to remember this topic. Migratory trace substances produced in one tissue and exert a specific effect of functional value on all other tissues is said to be hormone. Shall I tell you in a simple way? Migratory trace substances, they are produced in small quantities. They are capable of moving from one cell to another through xylem and phloem. They are extrinsic or intrinsic factors. They are intrinsic factors and intercellular because they are migratory, therefore intercellular. Intrinsic and intracellular factors controlling growth and development are genes. Intrinsic and intercellular is hormones. Why they said intercellular? Because they are capable of moving from one tissue to another through vasculature and exert a specific effect of functional values on all other tissues. So hormones need not be synthesized in every tissue, children. Even if it is synthesized in one tissue, it can move to other tissues through vasculature. That is the significance of hormones. All right, some scientist names. We'll come back to the scientist names in a minute. Darwin, he worked on canary grass and he reported auxins help in phototropism. Wendt, he worked on oat plants and he reported that curvature in oat coleoptiles is because of unequal distribution of auxin. I'm going to explain in a minute. Kurosawa, he reported a Bacone disease in rice is caused by the secretions of a fungus, Gibberella fusicori. Skoog, he is a scientist who isolated a chemical substance called zeatin from endosperm of maize. Scientific name of maize is Zea maize. So no prizes for guessing why he gave zeatin. And this zeatin along with auxin is going to cause rapid cell division in tobacco pit cells. Then Miller, this man dived into water, caught hold of a fish, and from the fish he isolated the DNA. He autoclaved, heated the DNA and got a nitrogen base which is capable of causing rapid cell division, and he called it as kinetin, and from herring's fish which is capable of causing cytokinesis. Cousins for the first time reported that ripe fruits are going to release ethylene, which cause the ripening of unripe bananas. Ripe oranges cause the ripening of unripe bananas. Richmond and Lang, not there in your book, cytokinins are going to delay senescence. That is the reason why it's called Richmond-Lang effect. When we go to individual hormones, we'll come back to this scientist. Please don't get worried with regard to it. I want to keep all the scientist names at one place. That is the reason I did it. Now, source of hormones. Auxins, they're first isolated from human urine. So, how can a plant hormone be present in animals? Well, naturally, animals, humans can also consume plants. That is one reason how and why auxins could be present in humans. Apart from that, even some amount of compounds similar to auxins. Auxin is not one chemical, it's a group of chemicals. Similar compounds can also be synthesized in humans, all right, from tryptophan, an amino acid. So therefore, auxin-like substances or auxins are first isolated from in human urine is an important point. Gibberellins, they are first isolated from a fungus, Gibberella fusicorae. Kinetin, it's first isolated from herring's fish, autoclaved DNA. Zeatin, it's first isolated from endosperm of maize. Ethylene was thought to cause ripening of fruits. Nice, easy way to remember. I kept all the scientist names and all that at one place. Chemical nature, auxins, indole acetic acid or indole butyric acid. Gibberellins, the most common gibberellin is GA3. Cytokinins, the most common natural cytokinin is zeatin. It is basic, means it is alkaline in nature. Ethylene, it is simple gaseous hormone, and abscisic acid is acidic. Both auxins and abscisic acid are acidic. And ethylene is a gaseous hormone and cytokinin is basic hormone. Precursor, children, precursor very important. For auxin, it is indole compounds. For gibberellin, it is terpenes. For cytokinins, it is purines. For ethylene, it is not there in your book, methionine. What is methionine? R is CH2, CH2S, CH3. That CH2, CH2 can get separated and form ethylene. Abscisic acid, this is a very important point. All right, carotenoids. A pigment is a precursor of a hormone. What is a pigment? What is a hormone? Pigment is carotenoid, hormone is abscisic acid. Where is the major site? Auxins are mostly synthesized in stem tips or coleoptile tips. Gibberellins are mostly synthesized in germinating seeds and nodes. Cytokinins are mostly synthesized in young root tips, shoot tips, and flowers. And ethylene is mostly synthesized in ripened fruits and senescent parts. All right. And the bioassay, it's a bioassay means what is the effect of minute concentration of hormone, all right, on plants is called bioassay test. It's like a litmus test. All right, auxin Avena curvature test. I'll tell you what is this. Gibberellin dwarf mutant variety test. I'll tell you what is this. Cytokinins tobacco pith culture test. We already discussed Coog culture tobacco pith cell in the presence of both auxins and cytokinins. Ethylene triple response growth and thinning. I'll explain this. Then abscisic acid, the bioassay test is it can cause seed dormancy and seed maturation. Apart from that, stomatal closure can also be the bioassay test. This is the chronology of work done. What did I do? I brought ethylene, all right, from the last and I kept here. So auxins, work was done first on auxin, then it was ethylene, then gibberellin, then cytokinin, then abscisic acid. All right, so I brought ethylene from the last and kept at second position. So therefore, easy way to remember. Next, a mass of undifferentiated tissue produced from internodal segment of tobacco Coog tissue culture. He added auxins and cytokinins. This, I don't know why I kept here. It's not needed here. Children, some physiological effects. Textbook says in the initial part itself that hormones cause different physiological effects. You will understand them once I complete, but because you already completed, so I can show you directly. Cell division, both auxins and cytokinins induce cell division, even gibberellins. Internodal elongation is by cell division and cell elongation only. Seed germination, it is gibberellins and ethylene. Gibberellins induce seed germination in barley, ethylene induce seed germination in peanuts. Pattern formation means how the arrangement of tissues, what is the plane of division, and auxins are responsible for pattern formation. Remember, this goes out of your memory. Phototropism and geotropism, the movement, the curvature of the stem and the root, all right, that is again auxins play an important role. Flowering, auxins, gibberellins. Auxins induce flowering in pineapple, gibberellins induce flowering by causing bolting, and ethylene induce flowering in mango, in pineapple, in cucumber. Very important. Abscission layer, not only abscisic acid, ethylene will induce abscission layer. Morphogenesis, the meaning of morphogenesis is development of root from callus and stem from callus, auxins and cytokinins. Fruit ripening, ethylene. It's too early, isn't it, to know all this. Once I go into the hormones, you'll be knowing it, but by this time you know all this. Auxins help in pattern formation, spatial organization of tissues and cells in plant, as auxins direct when and where cells divide and organize themselves during development. This is an important point. Then we'll come back to these lines a little later, children. Now, look here. When we start hormones, we'll pick 5 minutes for each hormones and get rid of it. What is the layout which we are going to adapt for each hormone? This is the perfect layout, I personally feel. Just listen. First, I talk about the scientists. Then I talked about what is the chemical name. Scientist here is Darwin and Wendt. Chemical name, indole acetic acid and naphthalene acetic acid. Then precursor, indole compounds, 3. 4, bioacid test, Wiener curvature test. 6, where it is mostly seen in coleoptile tips. And after this, root, stem, leaf, flower, fruit and seed. Isn't it looking simple, children? Scientist name, chemical nature, precursor, synthetic hormone, bioacid test, occurrence, then root, stem, leaf, flower, fruit and seed. Let us see. Let us put this table for all hormones and see whether it's more easier. Next, Darwin. For the first time, he talked about phototropism. Wendt, Wiener. Now, what Darwin said? Look here, I have shown here. All right. When light is falling on this side, children. All right. Auxins are sensitive to light. Look here, this is the place where auxins are present. Okay. This is the place where auxins are present. Stem tip or coleoptile tip. When light falls on it, then naturally auxins will be moving more towards that side. They go more toward the shaded side. Look here, this side, shaded side. When auxins move to the shaded side in large quantities, the shaded side surely will show more growth. And this illuminated side, light side, shows less growth. Then the shaded side which grow is going to look back. Why you are not growing? When it looks back, look here, it's looking back. That is bending towards light. This is said to be phototropism. Nicely I explained. Now you understand why this curvature? Because of unequal growth. Why unequal growth? Because of unequal distribution of auxin. Why unequal distribution of auxin? Because auxins are sensitive to light. Therefore, they move to the shaded side. More auxins on the shaded side. More growth on the shaded side. Therefore, it bends. Less growth on the illuminated side. More growth on the shaded, less. Therefore, it bends towards light. Phototropism. But if you cover this tip with any cap, then auxins are not moving because auxins are safe inside the cap. Then why they have to move to the shaded side? So therefore, there is no curvature. But if you cap it not at the tip but at the bottom, still curvature occur because auxins are present here, not at the base. Then Wendt, this is an important pun. What Wendt did, look here. He removed the coleoptile tip. What is the meaning of coleoptile? A small foliar sheath surrounding the plumule is said to be coleoptile. So don't get confused. Take it as stem tip, a part of the stem tip. Coleoptile tip he removed. And he kept it an agar block. Agar has got more imbibing capacity. It is going to absorb all the auxin. And what did he do? He kept an agar block on one side of the stump. Look here, he did not keep symmetrically on one side. Look here, this is the stump. He kept agar block here. So naturally, more auxins will move on this side, agar block side only. And there will be less auxins on this side. So naturally, there will be more growth on that side, all right, on this side, and there is less growth on this side. Then naturally, it is going to bend like this. So tell me, is it going to bending away from agar block? Yes, it's bending away from agar block. I repeat again, he removed the coleoptile tip and he placed an agar block. All right, agar block absorbed all the auxins. Up to here you understood. He kept agar block, look here, on one side, not symmetric. This is exactly on the middle is symmetric. So you look here, this is symmetric placement of it. If it is symmetric, auxins will be distributed everywhere. I'm sorry. You'll see a linear growth. Because he kept asymmetric on one side, more auxins will move only on this side, agar block side, less auxins. More growth on agar block side. Naturally, because there are more auxins. Less growth. This unequal growth causes curvature away from agar block side. This is said to be Avena curvature experiments. Nice. So that's about the scientist. Next, I told you natural auxin, indole acetic and indole butyric. Then I said synthetic auxin, 2,4-D and naphthalene acetic acid. Then I said bioacid test. This is Avena curvature test. He worked on oat plant. That point is important. Darwin worked on canary grass. Went worked on oat. Darwin in the presence of light. Went in the absence of light. Because he has to put light away. Otherwise, again, his experiment goes wayward. All right. That is an important point. Synthetic auxin is 2,4-D and naphthalene acetic acid. This is very important, children. 2,4-D. 2,4-dichlorophenoxyacetic acid. When Americans were unable to what is called win a war against Vietnam, because these Vietnamese soldiers are hiding behind the bushes and killing the Americans. You know what Americans did? They sprayed 2,4-D on the entire what is called area, such that all the dicot plants, all right, will die, such that they can have more visibility. All right, in spite of that, Americans lost. That's a different story, which that's not important for us. But 2,4-D is said to be what is called a synthetic hormone which eliminates dicot weeds in a monocot crop. Mature monocot crop. If the monocot crops are immature, if you spray 2,4-D, both dicots and monocots will die. So that is the word mature monocot crop. It is used. That word is important. Please see that you don't forget. Synthetic auxin, naphthalene acetic acid. It can also help in the root formation from stem tips, naphthalene acetic acid. But this is important. D for death, D for dicots. In that way you remember. 2,4-D cause the death of dicot weeds in a mature monocot crop. Bioacid test is Avena curvature. Precursor is indole compounds. We already discussed about this, human urine. So physiological effects. I told you we'll be going in a systematic order. First, we'll start with root. It helps in adventitious root formation at stem tips. So if I take a stem tip and if I dip it in naphthalene acetic acid, roots develop. What is the advantage? I can directly sow them and cultivate them more faster. Especially in horticulture and agriculture, this property that auxins can help in the development of adventitious roots will be extremely handy. All right. Then, auxins are going to induce apical dominance. Now, what is the meaning of apical dominance? Look here. This is the terminal bud, children. Concentrate. This is the axillary bud. We already discussed that auxins are more in the terminal bud. Yes or no? And what happens? This auxins from the terminal bud, they enter into the axillary bud. Then you may ask me, nice, no. Axillary bud also will be happy. No. This is the problem. When more amount of auxins enter into the axillary bud, already cytokinins are there in that place. All right, these auxins are going to fight against cytokinins and neutralize cytokinins, decrease the concentration of cytokinins. Already you know, cytokinins are going to help in branching more growth, all right, more lateral branching. So therefore, when auxins are going to decrease the concentration of cytokinins, neutralizing the effect of cytokinins, so naturally, cytokinin concentration decrease, there is no branching. When branching is prevented, only the terminal bud grows. This is called apical dominance. I repeat again. I have written here. What did I write? More auxins translocate into axillary buds, correct. It inhibits the growth of axillary bud by decreasing cytokinin activity, correct. Then there will be only terminal bud growing. This axillary bud doesn't grow. And that is called apical dominance. So if I am a tea plantation grower, then I am not very happy by seeing my plant growing tall. What I want is more branches. More branches, more leaves, more yield, I can gain more profits. Then what I do? I take a scissor and remove the terminal bud. So that is called decapitation. So decapitation of terminal bud. What does it do? It removes the source of auxin. So I am removing the source. When I remove this, I am going to remove the source of auxin. So naturally, more auxins will not enter. When more auxins will not enter, the cytokinins are safe. When cytokinins are safe here, they cause more branching and thereby it causes more bushy or hedge making. Removal of source of auxin. When I remove the tip, less auxins in the axillary buds, more cytokinins, one more way of saying, more growth in lateral buds, more bushy appearance, hedge making and tea plantations. Shall I put a dangerous question? What is the horticultural method responsible for hedge making? The answer is decapitation. What is the hormone which is responsible for hedge making? Be careful, it is cytokinin, not auxin. A hormone which is responsible for bushy appearance or for hedge breaking or more branches is cytokinins. Okay, be careful about that. Next, leaves, it prevents abscission layer at the base of young leaves. What is abscission layer, children? All right, at the base of leaf, if a suberized substance accumulate, at the base of the leaf, then more water and minerals cannot enter. When more water and minerals cannot enter, the leaves are going to fall. That abstracting layer, better word, abstracting layer is said to be abscission layer. So what auxins do is to remove this abscission layer such that these young leaves can grow. So it prevents abscission layer at the base of young leaves and promote at the base of old leaf. There is no evidence that cytokinins promote abscission layer at the base of old leaves. At the base of old leaves, they doesn't bother. They bother only on young. So that much you remember. Flowering in pineapple, it helps. Remember that. Fruit formation, seedless fruits. Parthenocarpy means seedless fruits in tomato. It also helps in more amount of lignin formation, such that the cells of xylem differentiate and mature. So it helps in lignification of walls. Therefore, it helps in xylem differentiation. That's about auxins, children, which is very simple now. You understood everything. Next, we go to gibberellins. A Japanese scientist called Kurosawa reported that the rice plants were growing abnormally tall because of the secretions of a fungus, Gibberella fusicorae. That is the reason why this chemical substance was given the name gibberellins, because it was first time isolated from a fungus. Natural gibberellin, GA1 to GA110. Most common type is GA3, remember that, 19 carbons. Precursor for gibberellins is terpenes. The bioacid test, dwarf mutant variety test, I'll tell you what is the meaning of that. Then roots, none. Of course, there may be some role, but in our textbook we don't have. Stem, it causes internodal elongation and thereby increase the sugarcane yield. So what you sell in sugarcane are the internodes. All right. So therefore, if it's more internal elongation sugarcane, then a little bit of genetics also if you bring here, children. Why the plant is tall? Your genetics tutor told by this time that a plant is tall because of its genes. Now what the gene will do? This dominant gene, all right, is going to produce a protein, and that protein is going to allow the synthesis of more amount of gibberellins. And these gibberellins cause cell division and elongation. When it causes cell division and elongation, especially in meristems, all right, there is more growth and more internodal elongation and the plant is tall. So now you understood why the plant is tall. A plant is tall because there is more growth. Why there is more growth? Because there are more gibberellins. Why there are more gibberellins? Because there is a protein synthesized from a gene, tall gene, which is capable for the synthesis of more amount of gibberellins. Then why a plant is dwarf? You studied about the meaning of a recessive gene. All right, it's a mutated gene, always a mutated one. And it is going to a mutated one. Then naturally, it doesn't produce a protein which is responsible for the synthesis of gibberellins. And as a result, the plant is dwarf. If such a dwarf plant, if I expose them to exogenous, means externally if I supply gibberellin, it grow tall like a normal plant because cell division, cell elongation is artificially induced, but produce only dwarf plants in the next generation because you are not changing the genotype of such plants. Did you understand dwarf mutant variety test? I told you a mutant variety which is dwarf can be made tall by the application of exogenous gibberellins. This is the bioacid test. Leaves we don't have. Flowers, children, bolting. What is the meaning of bolting? Increase in the internodal length of rosette plants. So you know what is called, this is the cabbage cauliflower. This is a bud. All right, these internodes are very short, but in the presence of gibberellins, these internodes become long. All right, that phenomena is said to be bolting. Then, proper size of fruits in grapes. Imagine if the stalk of the grape is short, then naturally all the fruits are kept very close to each other. But if the stalk is long, then all the fruits are kept very far away from each other. When all the fruits are kept very far away, every fruit can grow in its size. So therefore, proper size of fruit in grapes, and it also have a role in proper shape of fruit in apple. They become little oblong, and they're going to delay senescence, delay aging in fruit trees. When the aging process is delayed, naturally the market value also will be more, because the fruits are seen in some season which is off. All right, then we talk about seed. Gibberellins can also help in seed germination, children. Question is, how gibberellins induce seed germination? Here I'm going to show you. I think I have left a small part. This, I'll tell you how gibberellins induce seed germination. This is, all of you know, this is the endosperm, starch part. This is the aileron layer. All right, this is the starch part of endosperm. This is the embryo. All right, I'm not interested in this embryo. This is a single cotyledon epiblast, large cotyledon scutellum, radical, plumule, coleoptile, coleorhiza. This is the barley seed, monocot. All of you know that gibberellins are synthesized in embryo. It moves to aileron layer. Aileron layer is a protein layer. In the protein layer, alpha amylase is synthesized. This is a digestive enzyme. And this alpha amylase migrate to the starch part of endosperm and convert this starch into maltose. This alpha amylase convert sugars, polysaccharides into monosaccharides, such that these maltose can easily be absorbed by embryo. Embryo cannot absorb starch because it is insoluble in water. Therefore, starch is converted to maltose and the maltose is absorbed. So therefore, now when the embryo gets enough food and also get enough oxygen, germination of seed occur. This is the concept, children, which is very easy to remember. Let us see what you write. Gibberellins are produced in embryo, correct. It moves to aileron layer, correct. And in the aileron layer, it helps in the synthesis of amylase. These amylase moves to the starch part of endosperm and convert starch into maltose. All right, and embryo can absorb. It also helps in early seed production in pinus, conifers, by decreasing the vegetative phase. That also is important to remember. And this point is important. Abscisic acid and gibberellic acid are antagonistic with regard to seed dormancy and seed germination. Gibberellic acid induces seed germination, abscisic acid induces seed dormancy. When two hormones have an opposite effect, you call it as antagonistic. That's a very common point to remember. That's about gibberellins. We are about to wind up things, children. Now the next hormone is cytokinins. It is a cell division inducing hormone and which is basic in nature. First, we'll talk about the scientist. In Auxin, Darwin and Wendt. In gibberellin, Kurosawa. In cytokinin, Miller and Skoog. And we already discussed. Miller isolated kinetin from autoclaved DNA of herring's fish. And it's a purine derivative, 6-furfurylaminopurin, and it is capable of causing cytokinesis. Skoog, he reported that tobacco pit cells can be cultured in the presence of cytokinins. Remember, children, the source of cytokinins: coconut endosperm, maize endosperm, yeast extract, vascular extract, or DNA degraded product. All of them can be the source of cytokinin. So any of these sorts of cytokinin along with auxin is capable of causing tobacco pit cells to develop into plantlet. In tissue culture, it is used. Skoog, remember. Natural cytokinin is zeatin, not kinetin. Kinetin is an autoclaved product of DNA, so you cannot call it as natural cytokinin. Synthetic cytokinin is benzylaminopurin. We don't have in our syllabus. The bioacid test, tobacco pit culture test, we already discussed. Roots. This is very simple, children. All right, this is an explant. It first develops into callus, a group of cells. Then if I add more auxins and less cytokinins, more auxins, less cytokinins, this callus will develop into root. This callus develops into root. Then same, this is an explant. It develops into a callus. Now if I add more cytokinins and less auxins, more cytokinins and less auxins, then it develops into what is called shoot, stem with leaves, etc. So the development of root from callus is rhizogenesis. The development of shoot from callus is cotylogenesis. Put together is said to be morphogenesis. So remember, cytokinins along with auxins, both of them are hand in hand. So you can say them as complementary or synergistic with regard to morphogenesis. All right. More auxins, less cytokinins cause the development of a root from callus, and more cytokinins, less auxins help in the development of shoot from callus. Adventitious shoots. So if a shoot develops from plumule, you don't call them as adventitious. If a shoot develops from any structure other than plumule, here from callus, you call them as adventitious shoots. And cytokinins are going to eliminate apical dominance. We already discussed that. All right, more cytokinins cause more lateral growth. All right, bushy appearance. So therefore, auxins induce apical dominance, cytokinins eliminate apical dominance. Auxins and cytokinins are antagonistic with regard to apical dominance. Auxins and cytokinins are synergistic with regard to morphogenesis. Remember that. And in leaves, flowers and fruits, this point is important. In leaves, flowers and fruits, it causes nutrient mobilization. When more nutrients and more minerals enter into leaves, the leaves are going to retain their green color. The chloroplasts remain intact, and you can delay the senescence, aging. You cannot prevent aging, isn't it? You can only delay. Delay means you can delay the aging, yellowing of leaves. How can you delay yellowing of leaves? Only when chloroplasts remain intact. When will the chloroplasts remain intact? When there are more minerals and more water. And cytokinins are capable of causing rapid cell division, especially in cambium, allowing more xylem and phloem to be produced, such that more water and minerals will translocate into leaves, and these leaves are going to retain green color for long, and this process is said to be delaying senescence. Not only leaves, even flowers and fruits remain fresh for long. Vast life period in flowers and long shelf life period in vegetables is because of cytokinins, because they delay senescence, and this is said to be Richmond-Lang effect. Now children, then we talk about the last hormone, literally the last hormone. We are running out of time as well. All right, ethylene. Now ethylene has more inhibitory effects than promotory effects, you know that. It is said to be the simplest hormone. It is widely used hormone in horticulture and agriculture. Scientists, we already discussed, Cousins reported that ripe oranges release ethylene and cause the ripening of unripe bananas. So don't keep ripe fruit in a bag of unripe fruits. If you don't want unripe fruit to become ripe, all right, so don't do that. So if you keep one ripe fruit in the company of the other unripe fruit, the ripe fruits release ethylene and cause the ripening of remaining fruits. All right, so then precursor for that is methionine, not there in your book. Ethylene releasing chemical is ethiphone, which is an aqueous form. It can be readily absorbed and diffused slowly. Bioacid test is the fruit ripening, thinning and triple response growth. Site of synthesis, it is in senescent parts. Physiological effects, listen carefully children. Root, more number of root hairs. It helps in more number of root hairs. When there are more root hairs, more surface area, more absorption of water. Stem, it helps in internodal elongation of underwater rice plants. If the stem moves out of the water, it can grow and remain functional. Leaves, even petiole elongation of underwater rice plants. And very important, it induces that obstructing layer. Auxins prevent obstructing layer called abscission layer. Ethylene and abscisic acid induce abscission layer. When this abscission layer is there, it promotes premature leaf and fruit fall. A hormone which prevents premature leaf and fruit fall, auxins. A hormone which promotes premature leaf and fruit fall by inducing abscission layer is ethylene and abscisic acid. Flower, synchronous flowering in pineapple. All of you know pineapple is an aggregate fruit. all right, aggregate fruit. So all the flowers, all right, will develop into fruits. All the flowers. And you know, the entire fruit develops from many flowers. All the flowers will develop into one fruit. So if all the flowers develop at same time, all the fruits also will be formed at the same time, such that the entire fruit is more edible. So therefore, synchronous flowering is needed in pineapple, such that all the flowers will develop into fruit simultaneously, and that makes the fruit more viable, all right, more useful. More number of female flowers in cucumber, such that there is more yield. Flowering in mango. And it promotes abscission layer at the base of cotton, cherry and walnut flowers. Why? You may ask me why? Because look here children, competition. Imagine if a plant has got 100 flowers. Then the flowers will not grow properly in size because there is a lot of competition among 100. All the nutrients should be properly supplied to 100. Imagine if the same plant, the number of flowers is decreased from 100 to 50. Then naturally the 50 flowers can grow better. So means some flowers has to fall down. That is by inducing abscission layer. So is ethylene helping the plant? Absolutely. It is allowing the flowers to gain proper size and show its characteristics more properly by decreasing the competition, by eliminating some flowers. Thinning of cotton, cherry and walnut. It promotes fruit ripening. What is the difference between ripe fruit and unripe fruit? Ripe fruit, listen carefully, ripe fruit has more sugars, isn't it? So ethylene will help in the formation of sugars. Unripe fruit will have organic acids, tannins, etc. Ripe fruit will have more sugars. Ethylene have a role there. Have you seen unripe fruit? It is hard. Ripe fruit, it is soft. Why? Because the middle lamella is dissolved, because ethylene help in the formation of pectinase, which dissolve the middle lamella and allow all the cells to get separated. And if you have you observed a ripe fruit like mango, it is little warm. Why it's warm? Because of more respiration. So it means that fruit ripening is associated with increased respiratory rate, that is said to be respiratory climacteric. So therefore it promotes fruit ripening. How? By promoting more sugar formation, by causing the dissolution of middle lamella, and by increasing the respiratory rate, which is said to be respiratory climacteric. All right. Then does it promote abscission layer even at the base of fruits? Yes, not only flowers, fruits of cherry, cotton and walnut. It helps in seed germination in peanuts, remember that. But if they ask you a hormone which is famous for seed germination, gibberellins, all right, not only in barley, in majority of cereals, but for peanuts means you take this, sprouting of potato tubers. Triple response growth. I already discussed this triple response growth. Children, this seedling is going to form a hook. Why it forms a hook? You know the reason. When it forms a hook, it can easily go up without any resistance. This tip will get damaged if it grow like this. This is the soil. So if it grow like this, it's more easier. So that is apical hook formation. Then horizontal growth of dicot seedlings. Look here. If the seedlings have more what is called swelling, sorry, I'm talking about swelling. If the seedling, this axis is swollen, then it can easily what is called move up, all right, above the ground. So the swelling of an axis help the seedling to penetrate through the soil and move up because it has got more strength. Then horizontal growth of dicot seedlings. Imagine if there is any hard substance on the top of the soil, then the seedling have an ability of taking a horizontal root and then move up. So therefore, this is horizontal growth of dicot seedling. So in all these three, apical hook formation, horizontal growth of dicot seedlings and swelling of an axis, all these three put together is said to be triple response growth. Remember that? That is a bioacid test. Abscisic acid, there is absolutely nothing for me to talk about. It is stress hormone. If some of you are thinking that it is causing stress, it is absolutely wrong. It is going to relieve the plant from stress. How? Outside it's very hot. So stomata, if it is closing, then the plant can save water. So it causes stomatal closure. Outside it is very hot. The seed feels that it cannot germinate and develop into plant because the plant will die. Then abscisic acid says, don't get worried. Seed, you stay like that. I'm there. I'll keep you in the form of seed only for long, such that you can take rest till the rainy season. It induces dormancy. Is it doing a favor? Absolutely doing a favor. But it is causing temporary suspension of growth. No doubt it is causing suspension of growth, but it is helping the plant. Then by inducing dormancy, abscisic acid also helps the plant, helps the seed. It says that, look, I am causing a temporary suspension of growth. That will help you to mature properly and develop properly by taking some amount of time. Look here, this exam got postponed. Now you have got plenty of time, all right, to look into those aspects in which you are not good at. Isn't it, children? Please don't waste your time. It's an important time for all of you. Just like that, abscisic acid induce seed dormancy, thereby help in seed development and maturation. Common name, growth inhibitor or stress hormone. It is antagonistic, opposite to gibberellic acid. Scientists, 3 chemicals got purified and characterized: dormin, inhibitor B, and abscissin 2. All these 3 put together is said to be abscisic acid. Remember, hormone is not one chemical. Auxin is not one chemical, a group of chemicals. Just like that, all these group of chemicals put together forms abscisic acid. Precursor is carotenoid. The bioacid test, it causes stomatal closure and abscission layer formation. It is seen in old and mature parts. Physiological effects, I already told you. In stem and leaves, it causes stomatal closure, protects the plant from water stress, and it also causes leaf abscission and senescence. It induces dormancy in seed and allows the seed to mature and develop properly. Last 3 lines. Individualistic means if a hormone has a specific and unique role, like bolting is induced by gibberellin. This is individualistic. Morphogenesis. Both auxins and cytokinins join hands, all right, to do this job. That is said to be complementary or synergistic. More than one hormone doing a particular job is said to be complementary. Like seed germination, gibberellin, ethylene, you can say complementary. All right, that is one example for complementary. Apical dominance. Auxins and cytokinins act opposite to each other with regard to apical dominance. It is antagonistic. Gibberellins and abscisic acid, very important, act opposite to each other with regard to seed germination. Gibberellins induce seed germination, abscisic acid prevents seed germination. Finished. That completes the chapter called Plant Growth and Development. You have grown and developed totally and perfectly, all right, just like a plant in this chapter. I'm very happy now. We'll meet with one more topic tomorrow. Till then, keep reading.